A highly diverse group of organisms ranging from prokaryotic to eukaryotic forms. They were found in various ecosystems, including marine and with freshwater and terrestrial environments. Algae are classified into four main kingdoms: Eubacteria, Chromista, Planta, and Protozoa. This broad diversity has shaped a complex evolutionary history, particularly due to events like plastid loss, regain, and endosymbiosis. For a better understanding of their classification, algae are divided into 14 phyla and 63 classes, and with algae base offering an up-to-date and comprehensive database on their taxonomy. You can look on this base, it's very, very nice. With diversity is important because it underpins the wide range of function algae perform within an ecosystem, regulate key processes in this organism, including cell division, photosynthesis, and other biochemical processes, and then stress responses. In algae, brassinosteroids play a vital role in enhancing stress tolerance and contribute to biomass production. They were found across various plant species, but the impact of algal productivity is especially significant. These hormones are widely used in biotechnology, aquaculture, and pharmaceuticals due to their diverse and industrial application. The presence and role of brassinosteroids in algae offers existing potential to improving algal productivity. These hormones have been identified across two main kingdoms of algae, Chromista and Planta. They were found in several phyla, including Heteroconchophyta, Harophyta, and Chlorophyta. In the Heteroconchophyta phylum, brassinosteroids occur in species like Thalassiosira pseudonana from the Mediophyce class and Cystoseira mirica from the Phycophyce class. In Chlorophyta phylum, Clapsosormidium flaccidum from Clapsosormidiaceae class show a significant level of brassinosteroid, particularly brassinolide and castasterone. Additionally, within the Chlorophyta phylum, species such as Chlorella vulgaris, Acutodesmus obligus, and Chlamydomonas reinhardti also show notable brassinosteroid presence. Thalassiosira pseudonana contains significant levels of brassinolide, while Cystoseria mirica from Phaeophyce class only contain the compound 3-keto-24 epi 28 nor catasterone. In Chlorella vulgaris, on this algal model, I released many studies. It's a member of Treboxophyce class. Multiple brassinosteroids have been identified, including brassinolide, castasterone, and 6-deoxocastasterone. This species shown a particularly high concentration of brassinosteroids. Additionally, the Chlorophyce and Treboxophyce class trend to have a higher concentration of brassinosteroids compared to other algal groups. For example, in Chlorella vulgaris, we observed brasinolide at 70 picogram per gram fresh weight and castasterone at 470 picogram per gram fresh weight. Additionally, other brassinosteroids such as 6-deoxocastasterone, 6-deoxotifasterol, 6-deoxoteasterone are present in this species. On the other hand, in Acutodesmus obligus, we see lower concentration of brassinosteroid measured at 1.41 picogram per gram fresh weight and 28 homobrasinolide and only small concentration. This concentration, however, shows significant variability across algal species, which can depend on both the species itself and the environmental conditions they were exposed to. Brassinosteroids belong to a class of steroidal triterpenes, which are molecules made up to 27, 28, or 29 carbon atoms. The structure is composed of 3 terpene units or 6 isoprene units, which form the core structure. The brassinosteroids can be categorized into different skeletal structure types. For C27 brassinosteroids, such as those derived from cholesterol, for C29 brassinosteroid derived from campesterol, and for 29 brassinosteroid derived from beta-cytosterol. Each of these variations in structure plays a role in the activity of brassinosteroids in plants and especially in algae. The core structures can also vary based on specific chemical modification, such as 7-oxalactone, 6-hydroxy, 6-oxo, or 6-deoxy group found in different brassinosteroids like brasinolide, tiasterone, castasterone, and 6-deoxocastasterone. This slide presents the structure of brassinosteroids, only compounds which were detected in algae. The red color means that C27, the blue color is C28, and only 2 compounds belong to C29 brassinosteroids. Of course, this is a total number of carbon atoms in molecules. Only one from C27. It's a free keto 2022 epi 28 norcatasterone is occurs. Environmental factors such as salinity, stress, and exposure to heavy metals can significantly increase the levels of brassinosteroid in central algal species like Spirulina platensis. This stressor trigger the production of brassinosteroids, which help the alga cope with challenging environmental conditions. However, detecting brassinosteroid can be quite challenging due to the low concentration in algae. The methods used to detect these compounds are source-intensive, involving complex techniques such as LC-MS and or GCMS. There are, of course, also some gaps in research, mainly due to the high cost and limited available of algal samples, which makes large-scale studies on the occurrence of brassinosteroid more difficult. Nevertheless, overcoming this challenging is crucial to advancing our understanding of brassinosteroid and their role in algae. As I mentioned earlier on the previous lectures, the low concentration of algae in low concentration in algae brassinosteroids presents a significant challenge for detection. One of the most common methods which I recommended to you is liquid chromatography mass spectrometry. These techniques allows the high sensitivity and precision, separating compounds before analyzing them with mass spectrometry. Another, of course, which I used to many years ago, was gas chromatography mass spectrometry, GCMS, which is suitable for volatile derivatives of brassinosteroids. However, because brassinosteroids are not inherently volatile, they require derivatization before analysis, adding an extra layer of complexity. Despite these challenges, both LCMS or GCMS are crucial tools in the study of brassinosteroid, enabling us to quantify and understanding their presence in various algae species, of course. One promising method involves the use of enhanced LCMS techniques, which can increase sensitivity up to close 8,500 times, especially using isotopic labeling such as deuterium or tritium. This allows for precise quantification of brassinosteroid even at very low concentrations. However, one limitation of isotopic labeling is the possibility of isotopic exchange during the analysis, which can affect the accuracy of results. Another useful method is immunochemical assay. I didn't recommend this, but it is more cost-effective and suitable for real-time analysis. These assays use only some monoclonal or polyclonal antibodies to detect brassinosteroids, but their specificity can be lower due to the potential cross-reactions with other substances. Moreover, they may not be as sensitive with dealing with very low concentrations of brassinosteroids. While these techniques are effective, there is still room for further optimization to improve sensitivity and reduce limitation in brassinosteroids detection. Now let's take a look at some emerging techniques in brassinosteroids. They are not very popular. One such method is proteomic analysis, especially two-dimensional difference gel electrophoresis. The techniques can be used to identify proteins that are regulated by brassinosteroids. It's not, of course, techniques to detect a brassinosteroid and check the presence, the concentration. Its approach provides deeper insight into the cellular processes influenced by brassinosteroids. Another promising technique is isobaric tagging for relative and absolute quantification, which labels peptide for both relative and absolute quantification. Why this method offers great potential, they do come with some challenges. And for example, the data analysis process can be quite complex, and there is variability in plant proteome that could affect the results. Let's now summarize the key insight from emerging techniques in brassinosteroid detection. LC-MS remains the most commonly used methods for detecting brassinosteroid in algae. This method have proven to be effective, but emerging techniques like proteins, which I told 2 minutes ago, show promise to providing deeper insight into brassinosteroid-regulated proteins. It's very important, of course. Looking ahead, there are several future detections in brassinosteroid detection. One of the goals is to develop more cost-effective and sensitive methods for detecting brassinosteroids, especially particularly at low concentration. There is also the potential for integrating proteomic analysis with other detection techniques to create more comprehensive understanding of brassinosteroid function. The biosynthesis pathway of brassinosteroids begins with isoprene units, which form squalene and cycloartenol. These compounds then diverge into two main oxidation pathways: the early C22 oxidation pathway and the late C6 oxidation pathways. Both of these pathways ultimately lead to the formation of brassinosteroid and the most active brassinosteroid and the last compound in the pathways. Brassinolide is the most well-known brassinosteroids, not only present in plants, algae, and applied to studies. Similar to 24-epibrassinolide, in algae there is an interesting distinction. The precursor for brassinosteroid biosynthesis are produced via the non-mevalonate pathways in plastids, which is different from higher plants that synthesize from precursors in the cytoplasm. This unique feature of algal brassinosteroid biosynthesis is one of the key areas of study, as it could provide insight into the evolution of brassinosteroid pathways in different organisms. Among the important intermediates are compounds such as campesterol, campestanol, catasterone, and 6-deoxycastasterone, which are all precursors in the formation of brassinosteroids. There are also several critical enzymes that play a key role in the biosynthesis process. For example, it's DET2 reduce ergo-4-3N to ergostan 4-1. And a crucial step in the pathway. The next is dwarf 4 hydrolyzed brassinosteroid precursors at the position C22 and BR6OX1 enzyme adds a ketone group at the sixth position carbon. This enzyme regulates the formation of different types of brassinosteroids. There are totally 70, and ensure the proper production of compounds like brassinolide, which are, of course, essential for algae and plant development. This slide presents the brassinosteroids, the biosynthesis of brassinosteroids in higher plants and algae, where the name in the boxes indicate detection in both higher plants and algae, with a particular emphasis on algae. You can see on the end, I will show some references, and I recommend one paper where is this scheme present. As you can see, only one of the three pathways has been fully discovered, similar, of course, to the higher plants. The rest, C29 brassinosteroid pathways, is only partially understood, while the C27 brassinosteroid pathway is yet not to be discovered. It's a chance for us, it's a chance for you, of course. Brassinosteroid biosynthesis is heavily influenced by environmental stressors such as salinity and exposure to heavy metals. These stressors can trigger a change in biosynthetic pathways, causing algae to produce higher levels of brassinosteroids as a stress response. One of the fascinating aspects of brassinosteroids biosynthesis in algae is its pathway flexibility, which allows the organism to switch between different oxidation routes depending on the environmental conditions. Of course, more research is needed to uncover the specific enzyme activities and genetic regulation involved in these processes. It's not fully understood in algae, but vascular plants is. Brassinosteroids play a vital role in stimulating cell division and elongation process. They are crucial for the overall growth of algae. They also enhance the biosynthesis of key cellular components such as proteins, nucleic acids, saccharides, lipids, and all of which are necessary for healthy cell function and growth. In addition to supporting growth, brassinosteroids improve the net photosynthesis rate. Professor Shamsul Hayat present these reactions on his lectures, and brassinosteroid increase chlorophyll contents, both of which of course are essential for energy production in algae. Furthermore, brassinosteroid influence physiological responses by increasing cellular adaptation to stress and regulating carbohydrate assimilation and allocation within the cells. This function highlight the importance of brassinosteroids not only in promoting growth but also in the helping algae to adapt and thrive under challenging environmental conditions. Brassinosteroid boosts photosynthesis by increasing the level of chlorophyll A, chlorophyll B. This enhancement improves the energy efficiency in algae. Brassinosteroid also stimulate the production of carotenoids, especially lutein of astaxanthin, which is important, for example, in Hematococcus pluvialis. They also enhance the production of protein and extracellular saccharides, glucose, fructose, and others. Brassinosteroid also increase its phenomenon, the secretion. It's extracellular secretion of sugars and other organic compounds, for example, protein, which can be used by biofuel production and of course interact with other other organisms in aquatic environment. For optimal metabolic activity, brassinosteroids are most effective at the concentration which is present. This range of concentration has been found to significantly stimulate metabolic activity and enhance the production of these key metabolites. Under abiotic stress conditions such as salinity and temperature extremes, brassinosteroids help reduce the accumulation of reactive oxygen species in the cells. We know about reactive oxygen species that they can cause oxidative damage, and brassinosteroids play a crucial role in protecting algae from this damage. Similar in other plants, activating enzymatic oxidants and non-enzymatic antioxidants like glutathione, ascorbic acid, and other compounds. We know that this activation of antioxidant enzymes works synergistically with other phytohormones such as auxin and cytokinins to enhance the algal overstress resilience. The role of stress markers, not only in algae but also in plants, I presented earlier, so I omitted this knowledge. The combined action of brassinosteroids and other phytohormones help algae to be cope with environmental stresses, making, of course, brassinosteroids essential for maintaining cell integrity and function under challenging conditions. One of the most significant effects of brassinosteroids is their ability to increase cell division and the size. Chlorella vulgaris has a special big giant cell which divided normally from one mother cell into 4 daughter cells, but after brassinosteroid treatment, from one mother cells divided into 16 daughter cells. It's a really look fine when you can see biggest cells. It's a giant cell. It's fascinating. For example, of course, I provided in the studies in Chlorella vulgaris the application of brassinosteroid resulting doubling cell and size. The other key benefits of brassinosteroids is their role in lipid accumulation. These hormones manipulate lipid profiles, making them highly valuable for biofuels production and pharmaceutical application. One specific brassinosteroid, 24-epibrassinolide, has been shown to enhance both minor and major fatty acid production, which is crucial for optimizing lipid contents in algae. We know that Chlorella vulgaris is very popular as in medicine shops and as a supplement of our diet and in tabs or as a powder. Additionally, different brassinosteroid can be treated for specific metabolic outcomes. Compounds such as brassinolide or 24-epibrassinolide exhibit distinct effects on algal metabolism, showing the promising control over biomass production. What about interactions of brassinosteroid in other phytohormones? The synergistic interactions between brassinosteroids and, for example, auxin, cytokinins, gibberellin abscisic acid exist. One of the most significant interactions occur with auxins, particularly indole acetic acid. Brassinosteroids stimulate the expression of PIN genes, which are crucial for, as we know, for auxin transport. In turn, auxins enhance the endogenous level of brassinosteroid in algae is very fascinating and very important, and very stimulating to our next future studies on a molecular, of course, level, not metabolic or molecular level. Together, these two hormones promote increase protein, sugar, and pigments production. That's mean, of course, chlorophyll, carotenoid, xanthophylls, carotenes, alpha, beta carotenes. Another important interaction is with cytokinins such as trans-zaatins. When brassinosteroid and cytokinins work together, they enhance cell proliferation and improve metabolic outputs. This includes increase, of course, similar chlorophyll biosynthesis and trans saccharide production, both of which are vital for energy metabolism in algae. Additionally, brassinosteroids play a role in biosynthesis of abscisic acids, includes ABA production, which helps algae become more resilient to drought and salinity stress. This collaborative effect strengthens the ability of algae to survive in challenging environmental. These interactions show that brassinosteroids do not act alone, but rather function as a part of network complex, hormonal network complex that regulate algal growth, metabolism, and stress adaptation. Brassinosteroids also enhance production of carotenoids, such as astaxanthin, especially in Haematococcus pluvia. Is algae very useful in applications when acting synergy with yasmonic acid and salicylic acid. This interaction is particularly important for algae. Use in the production of pigments and astaxanthin for industrial and pharmaceuticals applications. Brassinosteroids regulate and improve antioxidant response, providing protection against abiotic stress such as extreme temperature and oxidative stress. These protection effects ensure better survival and physiological performance of algae under unfavorable conditions. Another crucial aspect of brassinosteroid is ability to regulate it, of course, biosynthesis, influence of auxin and cytokinase production, and other, of course, processes. What about the role of brassinosteroid in stress responses? Brassinosteroid mitigate effects of abiotic stresses. As you see, salinity, temperature, extreme low and higher temperatures, and heavy metal toxicity. Brassinosteroid, of course, act by reducing reactive oxygen species accumulation, and the key benefits is that protect cellular structures from oxidative damage and just stress tolerance. I told you about that earlier, that brassinosteroid increase activity of catalase superoxide dismutase ascorbate peroxidase, of course reduce lipid oxidation and maintains membrane integrity. The impact of on reactive oxygen species is lowers hydrogen peroxide and superoxide ion levels, and support, of course, cellular survival under these stresses. And how brassinosteroid can help algae cope with salinity and temperature stresses. In the case of salinity stresses, brassinosteroid help stabilize ion contents within the algal cells, preventing osmotic imbalance. They also boost the production of antioxidant compounds, which counter the harmful effects of reactant oxygen species generated by high salt concentration. This protection enables algae to maintain the growth and the metabolic function even in salinity stress. Under temperature stress, whether due to the extreme heat or cold, brassinosteroid help preserve chlorophyll levels and maintain carbohydrate metabolism. This ensures that algae can continue photosynthesis efficiency despite fluctuation in temperatures. Additionally, what is maybe new brassinosteroid enhance the production of abscisic acid, a hormone that play a key role in stress adaptation, further improving the algae resilience. What about heavy metals in the environment? We know that can be toxic to algae. Heavy metals can be toxic to algae, leading the oxidative damage, disturbing metabolism, and reduced growth. However, brassinosteroid play a role in mitigating these harmful effects. One of the primary metabolism is detection and protection. We know that brassinosteroids stimulate production of phytohalogens to detoxify heavy metals. Phytohalogens are molecules that bind to heavy metals ion and neutralize their toxicity. Earlier I present the biosynthesis of phytohalogens, so you know about it. This prevents heavy metals from accumulating at dangerous level inside algal cells. Additionally, brassinosteroid enhance antioxidant enzyme activity, which help counteract the oxidative stress caused by heavy metal exposure. Enzymes are the same, of course: catalase, superoxide dismutase, ascorbate peroxidase, which protect, of course, the cellular structures. As a result, algae treated with brassinosteroid maintain higher photosynthetic efficiency and overall growth even in polluted by heavy metals environment existed. This capability makes algae a promising candidate for phycoremediation and bioremediation, where they can be used clean up heavy metals polluted water through natural absorbing and detoxification processes. The graph summarizes the application of brassinosteroid in algae, showing anti-stress function. This graph comes from a publication which I published together with Professor Shamsul Hayat some years ago. The future research direction in brassinosteroid studies. particularly, of course, in algae, should focus on elucidating the C27 and brassinosteroid pathways. Of course, on 28 brassinosteroid pathways, it's not fully complete, but also two. Investigation that biosynthesis and functional role is, we know that remain undiscovered in algal cells. What else? Comparative analysis with higher plants, understanding the similarities and differences in brassinosteroid pathways and function between algae and higher plants, physiological and ecological function. We should explore the role brassinosteroids in algal growth, stress tolerance, and adaptation to aquatic environments. And then regulatory mechanism. It's still remain unclear. Identifying the molecular signaling pathways and the receptor mechanism of brassinosteroids in algae is very, very important. We don't know about anything of brassinosteroid signal transduction in algae. And the last biotechnological application, investigation how brassinosteroids can be used to enhance algal biomass production, biofuel generation, and stress resistance. And what else? Evolutionary perspective, omics-based approaches, utilizing genomics, transcriptomics, and metabolomics to uncover new brassinosteroid-related genes and pathways in algae. For example, enzyme activity, genetics regulation are needed to clarify they may be or pathways biosynthesis mechanism. We start this study on genomics. Finally, integrating modern technologies into brassinosteroids research could revolutionize the field of plant hormone. Genetic engineering could be used to enhance endogenous brassinosteroids in algae, leading to improved growth and resilience. Moreover, developing cost-effective methods for large-scale brassinosteroid production in algal cultivation could unlock new opportunities for biotechnology and industry. I told that brassinosteroid enhance lipid production for biofuel industry, nutraceuticals, pharmaceuticals, and wastewater management, which is very important. Another benefits, of course, that benefits, we have seen that how brassinosteroids play a crucial role. Phytohormones regulate fundamental processes like cell division, photosynthesis, and other. On the end, I would like to present the last summary of brassinosteroid occurrence and function in algae. I would like to recommend article with my PhD student on the same title, Around the brassinosteroids in algae, which was published in Algal Research. This is the references. The last Żeruń Jan, Bajgus Andrzej, Around the brassinosteroid in algae, Algal Research, 2025. And the next, of course, chapters published in Brassinosteroid Plant Growth and Development, edited by Shamsul Hayat, Mohammad Yusuf, Renubard Vai, and Andrzej Bajgus. It was in 2019. 6 years ago. Yeah. Thank you very much.